So sex hormones are a type of steroid hormone, and all steroid hormones are structurally related, so they can all cross-bind to some extent to each other's receptors. Progesterone, which is the first female sex hormone we're going to talk about, binds a progesterone receptor, and it's released by the ovaries into the bloodstream, so it has a systemic circulation. Testosterone, which is the first androgen, or male hormone, binds the androgen receptor at a very low affinity. It's also a systemic hormone. It's released by the testicles, and because it's systemic, it goes to every cell in the body. DHT, or dihydrotestosterone, is another androgen, and it's actually derived from testosterone and made within selective target cells. So as the testosterone goes through the bloodstream and is taken up by all cells, only those with the particular enzyme to make DHT are going to. It also binds the same receptor that testosterone does, the androgen receptor, but it's a very high affinity binding, and so it has some different effects than testosterone itself does. The last female hormone is estrogens, and we refer to a large variety of related compounds as estrogen, but in fact the compound that's made within the body at the highest concentration is estradiol. That's going to bind the estrogen receptor and is also systemic circulation. As you can look at the steroid backbone, and you might be able to remember that it has that structural component that looks a lot like glucocorticoids, mineralocorticoids, aldosterone, and all sorts of different other steroid hormones. So it's not that surprising that they can each bind at some affinity to each other's receptors. Although usually very little, we'll see that that's a big problem, especially with progesterones, because as we make more progestins, we can synthesize ones that have varied levels of binding to the other receptors. One of the most important things about steroid hormones in terms of their action is that they can be transcription factors. So when we talk about neuro, I talk about extracellular neurotransmitter type drugs that bind to receptors in the cell membrane and cause firing of neurons or non-firing of neurons. And we have a lot of drugs that are enzyme inhibitors. But for these, they're going to actually change the function of the cell in terms of what genes are produced. And so they're very important in telling the cell what type of cell it is and what type of function to create. So the steroid hormones, as we said, are systemic, so they're in the bloodstream, and they really enter every single cell. Most cells aren't going to have a receptor for that particular hormone, so only particular cell types are going to have that receptor, and say this is estrogen. When it enters cells that have estrogen receptors, it's going to bind to them, and that complex actually moves inside the nucleus. And that complex is actually the transcription factor. So it's going to bind to the DNA, open up the DNA at certain gene positions, and allow that DNA or that gene to be transcribed into RNA and then into protein. And so the attachment of this transcription factor complex is going to create a specific set of proteins being made, and those proteins are going to be specific to estrogen type cells or testosterone type cells specifically. So not only does this set of proteins and enzymes being made change the function of the cells, but a big thing for estrogens and androgens is that they are also growth promoters. And so they are causing expression of proteins and enzymes that are going to cause replication of the cells and tissue. So if you have an estrogen-sensitive tissue and you put estrogen on it, it's likely that it's going to replicate, and say if it's breast tissue, that breast will grow larger. So this is just a really quick conceptual restatement of that. You've got these different circulating hormones, testosterone, progesterone, estrogen, And as they go into cells, they're only going to act within cells that have their specific receptor. And that complex is going to then cause expression of specific genes, proteins, that are going to have the effect profile that we associate with either androgens, anabolic steroids, progesterones, or estrogens. And down here, DHT was the odd one because it starts with circulating testosterone, but as it goes into the cell, if the cell has that enzyme 5-alpha reductase, it's made into DHT, which binding to the androgen receptor has a different profile than testosterone does, and therefore causes a different set of proteins to be made. So as I said, a major role of them is cell growth modulators. And we see that as being important in tissue differentiation for sex organs in utero, but we also see it during puberty or during gender reassignment therapy for enlarging sex organs into for the ones that are sensitive to the hormone that you're looking at. And so the two that are that have a large growth promoter function are going to be estrogens and androgens. And estrogens are growth promoters at breast tissue, ovarian tissue, uterine endometrium. Those all have to do with female reproductive functions. They're also growth promoters at skin cells, and that's something that you see women tend to have a thicker skin cell layer, and so kind of have plumper, nice looking skin. And when they go into menopause, their skin becomes thinner. The vasculature also has estrogens, and women at high estrogen levels will make more blood vessels. And that makes sense if you think about pregnancy, because during pregnancy, your estrogen levels are really high, and you need to make more blood cells to go into the uterus, and so that you can have uterine and placental blood vascular development to give the blood supply to the fetus. And then also the estrogen receptors are in osteoblasts, and so those are the cells that make bone growth, so they deposit bone growth, whereas osteoclasts are the cells that break apart bone and degrade it. And there's always a kind of a balance between degradation of bone and rebuilding of bone that occurs all the time. And so when you think about women, when they hit menopause, tend to have a risk of osteoporosis. That's because they no longer have the estrogen that is mediating osteoblast formation. And then other effects of secondary sex characteristics that we see in women, pubic hair, and other effects like that. Androgens are growth promoters, so they cause cell division at muscle or skeletal muscle specifically. Men of the same family are going to have larger skeletal muscle development than women do. It causes growth promotion at genital tissue, at pubic hair, body hair, and also sebaceous glands, which actually comes in important when we talk about drug side effects, because the sebaceous glands make oils in the skin, and the more oil you make, the more likely you are to have acne. And so during the teenage years, when androgens are very high, both for women and men, although obviously more so for men, you have the strongest amount of acne, or the largest amount of acne. And we'll see that some of the oral birth control that women can take can have some androgen-like effects. And for some women, that birth control can actually increase acne. Progestins are the different ones here, and progestins are the class name for these drugs. Progesterone is the one that you see endogenously made, but progestins can refer to any of the related molecules in the body or contraceptives and drugs that we give. So these are actually growth inhibitors at some of the same tissue that estrogens act at. So they inhibit the growth of ovarian tissue, endometrial tissue, skin cells, and vasculature. So that's really important, because if you have estrogen only, you're going to have a lot of growth of all of those tissues, Whereas if you add progestins now, you'll still have growth at breast tissue and other types of tissue and osteoblasts, but you won't have it at the endometrial and ovarian tissues. So those can kind of compete with each other, and because they're high at different levels of the month, you're going to have some differentiation in the growth of those tissues at different times in a menstrual cycle. So the big picture here is, remember that estrogens and testosterones are growth promoters. They can enlarge sex organs, breasts and such during puberty, as well as during gender reassignment therapy. They maintain secondary sex characteristics like body hair, body odor. And unfortunately, if a cancer mutation occurs in a cell type that has that receptor, like breast cells with estrogen receptors, they will also speed cancer growth because their role is to increase proliferation. So they're not going to ever cause cancer. You have to have a mutation in order to cause a cancer. But if a cancer is present in a hormone-sensitive tissue, then that estrogen or androgen would cause it to grow faster. So the physiological effects of androgens depend on whether they're testosterone-like or whether they're DHT-like. And when you hear about on the news about anabolic steroids, they're specifically referring to steroids that have high effects that mimic testosterone itself. And anabolic effects are really about growth and metabolic function. And I can remember that by anabolic looking like metabolic. And that's how I remember those effects. DHT has what we call androgenic effects, and those have to do with sexual characteristics like sexual secondary sex organ differentiation, as well as the function such as spermatogenesis and things like that. So the anabolic effects are going to include things like a decreased cortisol production, which is a stress hormone, increased skeletal muscle mass, so men can build skeletal muscle more easily than women can. It stimulates red blood cell development, which can increase your hematocrit, but also because it's stimulating that development, increases oxygen supply and is very good in athletics, and causes increased bone length growth. And so that's what causes men to generally be taller than their female counterparts in the same family. So again, those anabolic effects, think metabolic. The androgenic effects or sexual characteristics are going to include receptors within the brain that have androgen, cells within the brain that have androgen receptors that increase sex drive and libido, can increase aggression, and those are going to be cells in those pathways that you can really see a difference when you take animals that don't have a sociological component, but also are really just driven by the hormones. And you can take rats, male rats, and take their testes, and then give them DHT. They will have huge increased sex drive to mate other rats, and they'll become extremely aggressive. And you can do the same with female rats. Give them DHT, and they have an increased sex drive and also become very aggressive. Another androgenic characteristic is high acne, which we talked about, the growth of beard and body hair growth in a masculine pattern, and then also male pattern baldness, if that's within your genetic history. Finally, we have the development and maintenance of male sex organs. So we all remember that in fetal development, we start out with female sex hormones, and it's the presence of androgens in the fetus that cause those organs to develop into testes and into the penis. And if that DHT is not present during development, female sex organs will be developed instead. Also, when men hit puberty, their sex organs are going to increase in size. We also need DHT for spermatogenesis, and it also increases the size of the prostate. So what we're looking at here when we think about increasing the size of the prostate, and we looked at the action of androgens as growth promoters, many types of prostate cancer are going to be androgen sensitive. And so men who have prostate cancer may very well be put on a drug that blocks testosterone DHT effects. So, of course, women have significant effects from estrogen and progesterone as well. And those, as I said, estrogen is often a growth promoter. Progestin can be a growth inhibitor in some of the tissues. And so we have for estrogenic effects, we have neuronal effects, again, because there are estrogen receptors in many cells. They can affect mood or depression, which can cause effects such as premenstrual dysphoric disorder, which is depression within the menstrual cycle. They can cause, for some women, migraines to become worse at certain points in their cycle. They change sleep patterns, and they increase pain sensitivity. And again, you can look at that with rats. You can take male rats, take off the testicles, give them high estrogen levels, and they'll all of a sudden have disrupted sleep cycles. They'll have increased sensitivity to pain. One of the really important things about estrogens is they're pro-clotting. So they're going to increase the clotting cascade. They also increase HDL over LDL, which is a good thing. They increase immune and inflammation pathways, which isn't always a good thing because that is the reason why women seem to have a lot more autoimmune disorders than men do. And often for women, those autoimmune disorders start to decrease in severity after menopause. We also see a huge set of tissues in which it's a growth promoter and is pro-proliferation. And that includes ovarian cells, the uterine endometrium, skin cells, breast, osteoblasts, pubic underarm hair, and vasculature. Progestin, on the other hand, is going to have also some complex mood effects, including their finding may be somewhat neuroprotective. And it's not sure how much that is, but they're actually looking at studies for giving progestins in high doses after people have a TBI or a stroke to see if that can keep cell death from occurring. The problem with progestins here is they are not opposing estrogens in a really important factor in which they are also pro-clotting. So given alone, progestins aren't as pro-clotting as estrogens are. So estrogens here have really high pro-clotting profile. Progestins only have moderate. But when you add them together, that gives you a very high pro-clotting instance. Progestins actually decrease the immune inflammation response, so they're opposing it. And as far as proliferation, they suppress proliferation of ovarian cells, uterine endometrium, and skin, but they don't suppress it in breast cells. And so when we see using progestins aren't going to increase the growth of breast cancers, but they will increase the growth of they are not going to affect the growth of breast cancers because they are not suppressing it, but they can help suppress the growth of ovarian cancer and uterine cancer. This horrible looking table is just kind of a rehashing of the drawings, which if you like tables might make more sense to you, and if not, you can ignore. It has all the same information talking about estrogen effects over here and progestin effects on the right, the types of cell proliferation that are increased with estrogen and opposed by progestin, the types that are increased by estrogen and not affected by progestin, the pro-clotting effects, and neuronal effects, etc. I kind of already mentioned that progestins also cross-bind to other steroid receptors. Now, all of the steroid hormones can cross-bind to other steroid receptors, but the progestins that we make in the lab tend to have a lot of problem with that. And so we would love to maybe make a progestin that is very specific and we can really control activity at just increasing progestin type effects, specific effects, but all of them that we have clinically are going to have some amount of cross-binding. And each synthetic progestin is going to have different types of cross-binding. So when you're picking a progestin to use clinically, you're going to have really different side effects depending on which progestin you pick, because one might have a lot of estrogen side effects, or another one might have a lot of androgen side effects, etc. Because it's so clinically important, there are some important progestins to know about and to know about their profiles, because that's really going to modulate a lot of therapeutic choice. So we consider having 4 generations of progestins, first, second, third, and fourth. And there are different schemes or tables that you might see that might have the third and fourth generation progestins have different drugs in those generations, because sometimes people classify them according to the time they were made as opposed to the structural class. The progestins here are given here. I'm only talking about 5 of them, but they are the most commonly used progestins clinically. And then we're going to look at their androgenic effects, their estrogenic effects, and aldosterone-like effects, which are salt and fluid balance. When I want to mention that when I talk about estrogenic effects, they're not really looking here at how much in the lab this particular progestin binds to an estrogen receptor. They're looking at a physiological outcome in human trials. And so the amount of estrogenicity it has is going to depend on what test they're looking at. So if they were looking at tests on breast growth, they might actually get a little bit different result than if they were looking at tests on skin growth or tests on clotting. So because it's one of the most important estrogenic risks that we have, we're looking at a clotting risk here, VTE, venous thromboembolism. But again, if you see slightly different results in a different table you're looking at, it's because they probably had a different outcome. So the first generation that's used very commonly is norethindrone. It started them all out. It's got a reasonable potency or binding activity to progestin receptors, but it has moderate androgenicity and it has low estrogenicity. So it doesn't have a lot of clotting risk, but it can cause for some women an increase in acne, increase in body hair, etc., maybe a little mustache. The second generation, they decided to try to synthesize something that had a better binding affinity to progestin receptors than norethindrone did. And so they were able to make a structure that binds progestin receptors really well, but they also ended up getting very, very high binding to androgen receptors. And so levonorgestrel is a very commonly used progestin, but it also has the highest risk of increasing acne, increasing body hair growth, etc. And many women don't like that side effect. It actually, though, while gaining the highest androgenicity, has the lowest estrogenicity. And so people who are worried about risk, stroke, levonorgestrel is probably the safest one to use. And look, if you remember, I said that estrogens have a higher risk for proclotting. Progestins do alone usually, but in combination, they have quite a bit more. Levonorgestrel may be the only progestin that actually may decrease clotting risk if given alone. But if given in combination, you still have, with an estrogen, you still have clotting risk. The third generation, desogestrel and norgestimate, have a moderate androgenicity because they tried to decrease that, but they went very high on the estrogenicity, and so now those are going to have the highest clotting risk of all. They then decided to do something completely different, and instead of making changes from the backbone drug that they had started with, they went ahead and tried something that was derived not from testosterones and physiological progestins and testosterones, but synthesized something that was very different. And what they were able to do was make something that's a very high affinity to progestin receptors, which they wanted, but actually binds to androgen receptors and inhibits them. So unlike all the other ones that activated androgen receptors to some extent, this is the only one that's actually anti-androgenic. And so women do often like drospirenone-containing oral contraceptives or hormonal contraceptives because they can actually decrease acne and decrease other types of symptoms that may be androgenic type symptoms such as PCOS, which you may talk about in a different lecture. That's polycystic ovarian syndrome, and it's the most common form of female infertility, and it's associated with fairly high testosterone levels. And so by taking an anti-androgen, often women can get some relief from those symptoms. So we also, drospirenone went down just to a medium estrogen risk, but also another thing that happened that was really interesting with this structure is it now started to bind aldosterone receptors. And it is anti-aldosterone, so it decreases salt and it decreases water retention. Therefore, it lowers blood pressure, which can be really good for some people. But unfortunately, it can also cause hyperkalemia because you have an increase in potassium retention in the body. So that can be a negative side effect. It's important to know the profiles of all of these 5 progestins because you will be choosing between them when looking at birth control choices for women. And this is just a activity that you can do to kind of quiz yourself. Is for testosterone, you can list some of the cell types and the effects for each, and do that for each of them as a review. Thank you.